कनेक्ट हो गया है, कुछ रहेगा इनका।
A very good afternoon to one and all connected here. A very good afternoon to one and all connected here. I am Pritika Murugesan, Head Event Coordinator, and on behalf of Center for Green Technology, I am greatly pleased to welcome you all to the 7th edition of Innovative Practices of Green Technology, organized by the Center for Green Technology, Institute of Chemical Technology, ICT, Mumbai, under the leadership of Professor Aniruddha B. Pandit, Vice Chancellor, Institute of Chemical Technology, and Professor Prakash D. Vaidya, Coordinator of Green Technology, Institute of Chemical Technology. Today, Team IPGT takes immense pleasure and privilege to witness the presence of our esteemed guest, Dr. Ashok Kumar, at our webinar. Good afternoon, one and all, starting with the session. Innovative Practices of Green Technology, IPGT, is an initiative to inculcate the importance of sustainable and green practices in industrial processes. It is an initiative taken by the Center for Green Technology at the Institute of Chemical Technology, which is a student-run organization, and aims to provide a platform for the exchange of technical views and information from the industrialists, academia, and the students in the domains of green technology and sustainability. Without any further ado, we would now start our session with the introduction of Dr. Ashok Kumar. Dr. Ashok Kumar carried out his PhD work at CDRI Lucknow under the supervision of Padma Shri Dr. Nityanand. This was followed by postdoctoral studies with the Professor Sir John Cornforth, CBE FRS, a chemistry Nobel laureate at the University of Sussex, England from 1981 to 1984. He started his career with Alchemy Research Center at ICI Bombay in 1985 and served Lupin Laboratories before taking up a job with Ipca Laboratories Mumbai, a leading pharma company, in the year 2000, and is presently heading the Center for Research and Development of the Ipca in the capacity of President. He is a co-author in more than 3 dozen papers published in peer-reviewed international journals, co-inventor in more than 150 patent applications, has presented scientific papers and keynotes in many national international conferences in addition to delivering invited talks on R&D management, idea management, innovation, role of common sense and philosophy in discovery research and many more such topics. It is indeed our greatest honor to witness your presence at IPGT, sir. Surely your professional outputs and experience will certainly enlighten us and leave a mark in our minds. Viewers are requested to submit their questions in the chat box of Google Meet or via live chat option in the YouTube Live. We are noting them down and will ask them by the end of this session. Sir will be delivering a talk on the topic, Unnatural Production of a Natural Product, Artemisinin. Sir, you may please begin with your talk.
Okay, thanks a lot for introduction, and thanks a lot to Professor Vaidya for inviting me to and also giving me an opportunity to speak to you all about some of the things we are engaged in at Ipca, and also something which we are not doing or we have not done, but they're very interesting. You just connect with the slides now. Slides. Is it slides? कौन सा? सर
ये ना
नहीं ये नहीं, पहले इसको स्लाइड शो है।
स्लाइड शो में करना रे
ऑब्वियसली
एक मिनट रुक
ये स्लाइड
दिस
पूरा इस्तेमाल नहीं था।
हाँ उधर जो दिख रहा उतना है
इतना ही है।
हाँ
Okay, so anyway, let me start with something which we are very familiar, and the topic related to green chemistry. All of you know that there are 12 principles. I don't need to go through it. But the point I have taken here I think it will take years for us to understand.
ये ना
अब मैं क्या करूं?
नहीं नेक्स्ट स्लाइड के लिए हाँ.
Right. I believe in something that why should we not question everything, whatever we do? So let me start by questioning the 12 principles we have with green chemistry. Can we think something different? And what I basically believe that if we really think, we always find out something different. And the purpose is that if we find something different, a different problem, then new solutions can also be provided. And that gives challenges and also something very, energetic to move ahead.
Excuse me, sir.
Yeah.
Sorry to interrupt you, but the slides are not visible.
Slides are not visible because they are not done properly, I believe.
Sir, I'll tell you, sir, I'll help you if you want.
Go back. आपको मालूम नहीं है, दूसरे को बुलाओ, इसको मालूम है.
नहीं इन लोग को दिख नहीं रहा है शेयर किया
It's not being shared here.
इन लोग को नहीं दिख रहा है
सर आपके गूगल मीट के स्क्रीन पे राइट बॉटम कॉर्नर पे रहेगा एक प्रेजेंट नाउ. आप प्रेजेंट नाउ पे क्लिक कीजिए.
प्रेजेंट नाउ इसमें का
यस एंड देन क्लिक ऑन योर एंटायर स्क्रीन. उसके बाद वहां पे एक स्क्रीन आएगा, उस पर क्लिक कीजिए और फिर क्लिक कीजिए शेयर. यस अभी आप अपने प्रेजेंटेशन पे जाइए और इसको फुल स्क्रीन कर दीजिए या फिर जैसे सही लगेगा इसको फुल स्क्रीन कर दीजिए.
ये full screen है अभी?
हाँ सर.
And this is for the movie.
हाँ.
Okay, so let me now speak again about this slide. This is talking about the 12 principles of green chemistry, which we are all very familiar with. What I say in this particular slide, let's question it, whether 12 principles cover everything, whatever it talks about green chemistry. Can we think beyond 12 principles and to find something different? And also, if we can find some problem which is different and not covered by 12 principles, then finding a solution for it may be very interesting. What I have written here, I think is a very general thing, that in general, we do not want to change status quo and we don't want to challenge anything, and we accept as it is whatever is being provided. But the reasons which possibly you call about the rejections of whatever we suggest and then leads to disappointment or non-acceptance of it can be avoided if we start thinking and challenging something. But in this particular case also, there are, I think, 12 principles. They are known for last, I think, 22 years, if I correctly remember, from 1998, and nobody has challenged it. I don't know whether I'm right to challenge, but I have tried it. And what I have basically come out of it, that a new principle I would like to add here, that why can't we do something from value, generate value from waste. So the new principle I am talking about is value from waste, and that too with commercial viability. You may possibly, as I have written in the first few lines, you may not accept it. You may find some explanations against what I am telling, but this is what I thought to share with you, and I will take you through a case study to prove my point. And I'm talking about an unnatural production of a natural product called Artemisinin. You just see the sequence here. There is something on the left side, sugar or molasses is a carbon source. And on the right side, there is a molecule, which is Artemisinin. Structure is given there. And there are two things written there, synthetic biology and synthetic chemistry. On the top of synthetic biology, there's something which may not be very clear to you, but this is nothing but yeast. In general, as everybody knows, yeast is not producing Artemisinin. What is being done here, that this Artemisinin is being produced by this yeast, and this yeast is genetically engineered to produce this product. And the latter part is done by synthetic chemistry. The product Artemisinin is a Nobel Prize actually chemical. I am not sure whether you know about it, but in 2015, if I correctly remember, some professor Yu from China. This is a lady, she got a Nobel Prize on this particular product because this was the major contribution coming from her. And this product is coming from a plant called Artemisia annua. It is shown on the left of the slide. This is a plant product and it is used in the first line therapy for malaria. Just to cover what exactly I talk about here, that malaria is affecting many people. So if you talk about 3.4 billion people in 92 countries, they are at risk. 228 million people in 2018 got actually malaria infected. Total deaths are in millions, and the most vulnerable people are in Africa. If we talk about the molecules which are being used for malaria treatment as a combination therapy, the left side is showing a derivative of artemisinin, it's artemether, and on the right side is the lumefantrine. So it's one of the combinations which is used for malaria treatment. Second chart shows artesunate, this is another. So the red one which you are seeing here, this is actually the keto group of the artemisinin is reduced and then it's derivatized. The derivatization is done basically for delivery purpose. There are two more molecules with artesunate mefloquine and also dihydroartemisinin, which is a reduced product for artemisinin with piperaquine, and also with sulfadoxine and pyrimethamine. So these are actually the products which are used as a first line therapy for malaria and widely used all over the world. If you talk about the biosynthesis of this particular product, this starts from farnesyl pyrophosphate, and this is anyway produced in yeast, but this particular product, when we talk about the conversion of the farnesyl pyrophosphate to amorphadiene, it doesn't happen in yeast. But in plant, which is Artemisia annua, it is getting converted to by oxidations, continuous oxidation to give DHAA, which is then cyclized in presence of UV light as shown on the right side. So the oxidation takes place first thing on the CH3 group to make the hydroxy, then aldehyde, then further this aldehyde is getting converted to acid, and this acid which is cyclized to give you artemisinin. There are many other products which are also forming in the plant, but this is the major product which we are interested in. The problem is that most of the product is actually getting accumulated in leaves of Artemisia annua, and the percentage which is available in the dry leaves is not more than 1.4%. So it can vary from 0.01% to 1.4% the dry weight. If you think of getting 6 kilograms of artemisinin, it requires 40 hectares of land. If you want a treatment for 100 million patients, you require Artemisia annua plantation at least 10,000 hectare land. And if you talk about all these things, it leads to many problems, price fluctuations, which can vary from $150 to $1,600 per kilogram. Short supply of the product, it can be a glut also. Forecasting is also a problem, and definitely the unsustainability in the long term. So what is the solution? So anyway, with problems like that, and the production of artemisinin is that critical, what exactly you can do? This slide shows a possible solution, which talks about a chemical synthesis. Well, we are all chemists. I think you also must be involved in organic chemistry. This is one of the syntheses I've taken from JACS in 2012, a beautiful synthesis, which starts from cyclohexanone, and by doing all sorts of modifications, it leads to artemisinin. This is not the only synthesis. There are many syntheses available for synthesizing artemisinin, but the fact is that if you really try to see the economic viability of this, when I did a little bit of workup on that what exactly may be the cost about this, it turns out to be around 5 lakh per kilogram. So if you think of a product like artemisinin, which is used in mass consumption, a 5 lakh a kilogram, you can understand it's not possible to use it. So what is the solution? And one of the dream solutions one can think of, that if plant is not feasible, chemical synthesis is not feasible, then fermentation-based production can be definitely a very interesting thing. And many things are important here. First thing is the green chemistry. So anything you do with fermentation, it stands in the green chemistry. It avoids dependency on plant, Artemisia annua in this case, and you don't need a land. So if you don't need a land, if you don't need plant, if you don't generate waste, I think all these things are possible only when we talk something very fantastic, and fermentation can definitely do it. But unfortunately, it does not happen because none of the fermentation process, it is known to be produced. Yeast is not producing, none of other bacteria is also producing it. So if it is not possible, then is there any way out? And let me show you something very interesting. There is some professor called Kisseling. He is in the Bay Area in San Francisco. What he did, he made it possible by doing genetically engineered.
I'm helping people stay connected with others near and far.
By genetically engineering a yeast to produce a different type of yeast, which contains a total metabolic pathway of Artemisia annua. And this yeast, using molasses or sugar, produces artemisinic acid, a precursor for artemisinin. And the most interesting thing is not to just produce it. In the paper 2013, it was published in Nature. They had published it gives 25 grams per liter. The recent information which I have, they now talk about that it can be done in 40 grams per liter. So you can understand that which was never possible by something like yeast or by any other bacteria, now it is possible to produce artemisinin at a very high titer, 25 to 40 grams per liter. If anybody is interested in this particular paper, this is a pattern et al. Nature. I have given the reference is 2013 paper. And if we talk about how it has happened, so if you talk about yeast contains this particular system. So acetyl coenzyme A through mevalonate pathway, it gives FPP. That I think all of you must be aware if you are knowing about the cholesterol biosynthesis, but rest of the things are not. So it does not do transformation of FPP to amorphadiene. If we see about the marvelous, I think, technology developed here, the pathway, whatever is there, it produces only a micro grams of the product. This has been overexpressed using heterologous gene expressions. This heterologous is basically means that any system which is not having those genes, and these genes are taken from outside and incorporated in that bacteria or yeast, this is called heterologous expression. And then discovering quite few things like cytochrome, plant dehydrogenase, which were not there. And as I mentioned to you, the conversion of acetyl coenzyme A to amorphadiene using a particular ATS, it was also not there. So yeast, which was not producing that compound, is now able to produce by putting this total genetic circuit in the yeast to give the final product, which is artemisinic acid. And then it is converted to artemisinin by a chemical means. So this is the first stage where you get artemisinic acid out. As I showed in the last slide, it gives 25 to 40 grams of artemisinic acid, and then it is converted by chemistry. So you are simply taking this is artemisinic acid, you reduce the double bond, normal chemistry, again you take this to esterify, and then you are doing a reaction with singlet oxygen to introduce a hydroxy peroxide group. This then it goes through Hock rearrangement and cleavage. If you are interested in the chemistry, it's very simple chemistry. You would just do the rearrangement like this, and it cleaves back, giving a 7-membered ring, which is a ketal, hemiketal. This is so unstable, cleaves back. Then you are reacting it with tertiary, triplet oxygen to introduce a OH group here, which comes here, and then it automatically cyclizes to give the artemisinin. And here, the overall yields reported here are around 40 to 45%. The same process, which was developed by Ameris, by the group of this Padden and Kisseling, was taken by a company called Sanofi. It's a multinational company because they had been working together and they had put a lot of money in development of that one. Did this development, it's the same thing I had shown you in the last slide, by doing some little bit of modifications to get it practically done using a light catalyzed reaction to get artemisinin in reasonably good yields, about 55%. They have actually introduced this product, this semi-synthetic artemisinin, in the market, but unfortunately it was not very attractive from economical point of view. It was coming around $500 a kilogram, which was definitely too much to have when you are talking about this product available in less than $200 a kilogram. Now you may be wondering that what exactly is my role and what Ipca has to do with this story. As I told you that this is an anti-malarial, and Ipca is the largest producer of anti-malarials in the world. And what happened that we visited Emirates. This is the company who had ultimately made it happen. And we were interacting with the CEO. And he mentioned two things. That production of artemisinic acid by fermentation yields a lot of amorphadiene. Means the amorphadiene produced in the whole process is almost double than what is required by artemisinic acid. It's a huge amount. And in case if we need, it is possible to produce in a very large amount. Means 120 grams a liter can be produced if interested. So what I thought, that if amorphadiene is available, since it has all the stereochemical requirements for producing artemisinin, it's not a bad idea to think of starting from it. And it can definitely lead to a viable synthesis of artemisinin and possibly the required target, which is the USD $100 a kilogram, if everything goes all right. As any chemist or anybody who starts with literature search, we did the literature search, and what we found out that whatever I was thinking, Emirates has already explored everything. I've just given you two approaches here. So what they had done, they had started with amorphadiene. They did the epoxidation to get this side chain epoxidase, and then converting, as I told you earlier, to alcohol, ester, acid, and all sorts of things. Sorry, aldehyde, acid, then ester, then doing the same Hawk rearrangement and getting artemisinin. But the raw material cost, because one thing you see the first step. In this first step, the exoepoxide, it is giving 34% yield. It's not very good as far as any chemistry is concerned. And when I calculated the RMC of this, it turns to be around 5 lakhs per kilogram. Yeah, and this is another synthesis. What they have done here, that they have used hydroporation as one of the step, the main step, getting the corresponding alcohol, although this is also giving another isomer. Then again going with the same chemistry, aldehyde, then getting the corresponding acid, ester, and then singlet oxygen reaction, and then triplet oxygen reaction, and ultimately getting this. But here also, the cost of 9 BBN, which is giving reasonably good selectivity here, is also coming around 2 lakh a kilogram, which is again too high to make this product anyway reachable to the common mass. So what actually I thought, and I discussed with them, that is it possible for you to provide me with this product, amorphadiene, to do some studies on that? And I believe that there is some scope. At the moment, one thing let me tell you here, I was not even knowing the structure of amorphadiene at that time. But they agreed that, okay, yes, we can supply. But when I interacted with the scientists there, what they say, there is nothing much left in this transformation, because whatever was possible, we have tried. And if you think to produce this $100 a kilogram, I think it's only a dream, it's not possible. But what I thought, let's take it as a challenge and not to accept failure without making efforts. Because I personally feel that it's always possible to find a viable solution to a problem, and we can do it. So this was exactly the thought came in my mind and requested them to supply Amorphadiene to start working on it. Let me now share what actually we did with Ipca. Usually the organic chemists try all sorts of reactions. We tried the same thing as everybody might have tried earlier, that how we can functionalize the side chain. Either it's OH group, either it's XCLBr, or it is epoxy, which they had already tried, or directly oxidizing it to get the required artemisinic acid. But you can see that none of the requirement was really near the requirement, which was 100% or at least 80-90%, where you can think of proceeding with that. We also tried another reaction, which is a very common reaction using MCPBA, metachloroperbenzoic acid, is the most common reagent available in any laboratory. It's a very costly thing to start for epoxidation, but when we didn't find anything as a ray of light to think of any new idea, we thought, okay, let's try this out also, let's see what happens. So when we tried epoxidation using MCPBA gave 3 products. One of the product, which was definitely not wanted, this was the endo epoxide, and 2 of the products, mainly 15%, which is coming as a diepoxide, and about 5 and less than 5% of the epoxidation was taking place at the place which anybody would have been interested, because this is the place where you want to do the functionalization. So looking at this particular sequence of reaction where the results were in front of us, we thought something that is very interesting, that we are seeing reasonably very high selectivity as far as the endo is concerned. If we avoid the over-oxidation, because this is the result of over-oxidation, if we are having more of the reagent, it leads to epoxidation at both the places. But this is not very selective for this. It is possible to really think of making very highly selective endo epoxidation and possibly a very high or good yield of this product. So the proposition was as simple as that. That it may be, it's just a hypothesis, or seeing a dream, that it may be interesting to, if we can get endo peroxide, and that comes in 100% selectivity and also in 100% yield. If conditions are developed to functionalize this endo epoxy AD, means the epoxy AD which is having the endo peroxide without touching the epoxide. I had never seen many examples where the epoxide has been used for doing many chemical reactions here and there and is not getting cleaved. Everybody knows there's a very strained ring, and even if you touch with a small mild acid, anything, it gets cleaved. And if the whole thing works with the cheap reagents and those catalysts which are very cheap or recyclable, maybe we can think of developing economical synthesis. So it was all a dream actually, because based on whatever results we had in hand, it was really not possible to think beyond that. But we thought, okay, let's see the dream and let's see that what we can do with it. The same thing I'm showing in a pictorial form. So this is the Amorphadiene. What was thought that if we get almost 100% yield of this endo epoxide, then we can try to do functionalization. This double bond is gone. There's only one active group, and then active methyl can be activated. Either we convert to chloro, bromo, or hydroxy, depending on the reactions conditions. And this can then be oxidized further to get the required epoxy artemisinic acid. And if we cleave off this epoxide after making it, it may be possible that we may get artemisinic acid, the intermediate which is being produced by the plant, by the fermentation process. And let me tell you the aha moment, that when we tried many things, but ultimately when we got this reaction done with hydrogen peroxide and SUH, which you can call green reagents altogether, we got almost 98% selectivity and yield of this product. So the great thing happened to start with was the endo epoxidation giving very high selectivity and very high yield. And when you get something done, it starts again, it gives you, sorry, I think just, नहीं ये बैक नहीं जा रहा है, कहीं और जा रहा है। IIT. Right. Sorry. So now this was thought of that, okay, this is great. If we are getting very high selectivity and yield of this product, then we can definitely functionalize with maybe if some simple reagent like bleach, and then further oxidations can lead to the required product. And practically it actually happened that when we used bleach, it could convert this to chloro compound, and using very simple reagent like DMSO, we could oxidize it to get the corresponding aldehyde, and with sodium chloride, we could oxidize to get the corresponding acid. So this was really fantastic that the things were very smooth, and we were very excited to think that the simplicity of the chemistry and also the use of the reagents which are very cheap, we may definitely get the target achieved. यार ये तो बड़ा प्रॉब्लम है यार. I think I don't know how to. Yes, that's right. But never you get everything done unless you have some setbacks, and only setbacks give you something possibly fantastic also. But when we tried the removal of the epoxy group to get the corresponding double bond back using lithium, which was a very simple reaction, and actually before we thought of this scheme, we had tried epoxidation of amorphadiene, as I told you earlier, and we also recovered 100% amorphadiene by de-epoxidation. So this reaction was already tried with lithium to work beautifully there. So we were confident that this reaction is likely to work, but unfortunately, I think it gave all sorts of possible products, and it was a mixture of products not workable to give the second stage. I think this is really a problem. पहले ठीक चल रहा था, पता नहीं क्या हो गया. But anyway, all problems come with some solutions. So what we tried, that okay, it's not working that way that we can remove this epoxy group. Definitely there is another possibility. Can we reduce this double bond? And we do simple reaction using hydrogen hydrate. This is again a green chemical. When you do hydrogen transfer using hydrogen, it gives you nothing but nitrogen. And this really worked beautifully. So what we really did not get in the first case by simply deoxidation, we got the dihydroartemisinic acid. That was ultimately the requirement. And when we subjected it to lithium epoxidation, it gave the corresponding double bond in a very high yield. So let me tell you that what exactly is it in the total summary. So we started with amorphadiene. We did the epoxidation of the unwanted epoxide, and all through it was used as a protecting group, now you can say, to get the final product. So epoxidation and then all the things which I have already mentioned. Here again we tried TCCA, this is a trichloro isocyanuric acid. It contains 3 chlorines and it can be used for 1/3 of the reagent, and it can be recycled and reused. So this was used. Again, we got very high yields and very good results. Same thing, whatever we tried, it worked. But only one thing which we did not get was anyway of not much importance, because ultimately we got DHA, that was the required acid for next step. And this is where this shows the conversion of dihydroartemisinic acid, as I have already discussed with you. And this is the mechanism of the Hawk reaction and the Hawk rearrangement to give the corresponding artemisinin. This is the introduction of this triplet oxygen here. This is the singlet oxygen reaction here. And let me tell you, this work we have published in OPRD. Oh my goodness. And we got an overall yield of 64%. If the RMC, as it was told by Amaris, of artemisinic acid is around, sorry, it's not artemisinic acid, amorphadiene, $30 a kilogram, we are able to get artemisinin in less than $100 a kilogram. And this is another thing which came out because this work was appreciated. And in the CPHI at Madrid in 2018, we got an excellence in pharma API development, and this is the shield which I am showing you here. So the work was appreciated as a green solution for this, and it was a practical solution which led to this sort of reward. Let me talk about the learning and a message to young scientists based on what I have just talked about. I think the two statements I have made. One is that the feat achieved by Professor Kisseling's team is unprecedented and obviously the marvel of technology. Whatever he has done, it was never there. Whatever he thought, it was not present. Whatever he has done, it was never believed. So whatever has been done in the practical sense, making something in such a huge titer and quantity using this technology is one of the most wonderful practical achievement I can tell. And whatever work we have done is also not that bad. At least it has made something possible which was not visible at that time. So it delivers a message. And the message is that if anyone has faith and confidence in self, the understanding of the problem, whatever you are doing, you are not afraid of failures, and you have the ability to persevere, means you can sustain all sorts of failures, problems which are going to come, whatever you are going to try. And if it is a tough target, there will be many problems. I think there is nothing which cannot be achieved. So this is my message to the young scientists, that try to have confidence in yourself, try to do something different, try to make a difference to yourself as well as whatever you are doing. And I think this is the only way to enjoy life and possibly chemistry, or specifically green chemistry. Thank you very much.
Thank you so much, sir. Thank you, sir. It was indeed a very illuminating talk and quite motivating too. Thank you, sir. I'm sure all the students, industrialists, and participants would have tremendously benefited from your deliberation here at IPGT. Thank you, sir. Vaidya, sir, if you may please. Sir, you're not audible, sir.
Not yet.
No, sir.
You have to, I think.
No, sir, you're not audible. I think if you...
Mic is switched off, I think.
And participants are requested to please leave any questions if you have any in the comment chat box, so that they can be delegated to the speaker.
Is there any question in this? I don't know.
Hello, am I audible now?
Yes, sir. Yes, sir. Yes, sir.
First of all, let me thank Dr. Ashok Kumar so much for giving such a nice lecture to our students. I have 2 or 3 questions for you, sir, and then I can just brief you about this course and this initiative.
Sure.
Sir, the first question is, you are a veteran in research and development, and our students are at the master's level, and they will be doing similar work in research and development.
Right.
So the first question is, now because they are learning about green technology and sustainability and so on. So
Right.
suppose there is a product that is to be made, and there are 3 or 4 ways of making that. So the question is, how to assess which method is most sustainable? Is there any way to assess that?
No, you see, this is all mental. That you have to simply see that what are the various possibilities and what is the sustainability you are looking at. So suppose if you are talking about a green chemistry and what you are using is not green, it's not recoverable, and the cost is going to be on the higher side. If you are worried about cost, then obviously these are the things to be seen. But I think more important is that the understanding of whatever the problem is and a wide knowledge, because it's not only one thing. You need to know a bit of chemistry, you need to know what are the possibilities existing. You also need to understand the that the more important is how you are really going to connect few things which are not connected. So creativity is more important because anything which you want to solve differently, that solution is not available. So you cannot simply take it and copy it. So you need to really, I think one has to read more. One has to read many things. One has to try to absorb, put it together, make cross connections and all sorts of fermentation combinations possible. To do that, this is the way to grow, I think. That's a simple thing.
Yeah, sir. My second question is: there are so many ideas while working in a lab. There are so many ideas. Some of them will lead to successful commercialization, and some will not. So, how to differentiate, and what is this journey from idea to commercialization?
You see, I don't think that at your level, particularly I'm talking about the student level.
Yes.
They should not worry too much about the commercialization.
Okay.
What they should worry about, can they do something which they feel is different, nobody has done it. So it's more an innovation or maybe something creative. If you do that, then maybe a person like me will take your idea and make it commercially viable. So a student is supposed to come out with some idea which is not existing.
Yes.
It should not be left to me. You see, if I am doing this work where I am doing some innovations, I think it would have been better it was done at your end, and I am making it commercially viable by making some modifications there. So for students, my personal suggestion is that let us not worry about that we want to make it commercial. They should not worry about knowing what industry wants to do, because what is happening in India, at least all over the world, we are talking about skill development. And that skill development actually is perceived that if a student is going to industry, and they are tuned for industrial requirement, means they are learning something which can be directly utilized in industry, then they are actually successful or qualified. I simply discard this idea. I personally feel that the student at the university level should do what university is supposed to teach them. New ideas, new way to think, creativity or something. They should not worry about the success as far as the commercialization is concerned. That's my personal way to look at it. Okay. Though I am working in industry.
Yeah. One more question that is again for the students.
Sure.
Now, these students, they will go to the industry tomorrow. Right. And they come from various disciplines. Some are chemists, some are engineers, some are technologists. So they have to deal with all type of people. For example, engineer has to deal with the chemistry expert, the technology expert, the marketing expert. So they also have to maybe look at what the customer needs and so on. So are there any some special skills in addition to research that they should be having today in today's industry?
You see, the answer which I had given you the second question, let me repeat it again. You see, I don't know what, but let me tell you, when I recruit a person, I really never ask one question that what you can do in industry. I'm not interested in getting any person who is coming with some experience which I want to use. I'm not interested. In my department, at least in my group, most of those people, they are contributing. They are all different types. I have not recruited because somebody has done something and I want to repeat it. So I have an engineer, I have a chemist, I have a pharmacologist, I have engineer. So all sorts of permutation combinations I have. This is the role of the leader. Okay. That how he is using and making these people becoming better and better and putting things together in such a way that it can make a product. But an engineer does not need to know chemistry on the very first day, but an engineer is already supposed to know a little bit of chemistry. A chemist is already supposed to know a little bit of engineering also, because all curriculum at university level, all sorts of permutation combinations are there, not in exclusive way. So whatever experience a person is earning at a university level, it's perfectly okay. As far as myself is concerned, I am supposed to now use that particular activity, his creativity or his expertise, how I can put it together in such a way that it can give me something different which I want. Yeah. So no need to think different from others' angle. It should be thought different from the person's angle, from university angle, from learning angle, from creativity angle. and that's good enough.
Correct. Sir, your CV is so impressive. So on a lighter note, what
Yeah.
is the secret of writing so many patents?
Oh, patents. Anything we do, we start with a simple philosophy, as I actually mentioned in my talk. What difference we can make here? As you see, somehow I have a firm belief. Let's not copy because whole India is copying. You see, we are simply enjoying copying and we feel very great about ourselves that we are doing a wonderful job.
Correct.
You see, everything what we do in India is copying.
Yeah.
It's very unfortunate.
Yeah.
Somehow, I have some gene just like Artemisia and your gene in East. I have some gene which somehow react to the copying, and that's why whatever I have actually done, at least something was different there. I have never repeated anything, and that's the problem. I cannot repeat and stick with the same problem, so I keep on moving ahead.
Yeah.
So I think that's all. But it's a gene problem. I think I really don't know beyond that. Yes.
Today, the government of India is taking a lot of initiatives to reduce export dependency. So there is a lot of emphasis on, for example, make in India. So how does your organization take this ahead, take this forward? What are your...
As far as the industries are concerned, my organization, we are anyway doing the same thing what is being asked for, make in India or made in India. It's not made in India, but make in India into sense. So most of the things are coming from, the innovators or those they have really developed the product or processes. And we are anyway making it in India and delivering it. And then we are exporting and marketing in India also. So I think from that angle, we are definitely, already doing it. But I think there is a more to do and a lot more can be done. So I will be more interested in talking about made in India than make in India actually. And this is one of the example what I have just given to you that we have made a difference, whether it's good or bad, big or small, it's not important. And whatever we have done till today, I think at least in my total career, I think every day there is something new happening. And there are many things we have done. I really feel good about all this.
Yeah. Sir, thank you for your very patient answers. Now, let me give you a very brief introduction about this program. So
Please.
all of us belong to the MTech program in green technology.
Okay.
This program was started at ICT in 2010. So it's almost 10 years. This is a master's program. It's a 2 years program started by Professor G.D. Yadav, our first and then Vice Chancellor. So at that time, it was his idea. It started with a Center for Green Technology in 2009. And in 2010, we started admitting students for MTech as well as PhD in green technology. So our students are a mix. They are a mix of B.Pharm. They are a mix of B.Tech. And they are a mix of BE chemical engineers. And then they are also biotechnologists. Some of them are also from chemistry. So it's a mixed class. Every year, 30 students are admitted for masters, and about 5 students are admitted for green technology. So these students, they first year, it is by papers. So they sit in the classroom and learn and appear for exams and so on. And in the second year, they go to industry for some implant training, short training, maybe up to 3 months. And thereafter, they conduct research in our labs. So now this research is also in different fields, depending on the choice of the students. So some of them work in pharmacy, some of them work in chemical engineering, some work in chemical technology, some work in biotechnology, others work in chemistry, and so on. So it's a good mix. And so far, it's going on very well. There is much response to this course. And we look forward to listening to people like you so that we come to know what exactly is expected today of our students. It is always good to listen to people from the industry. Students get an idea about what is really happening in the outside world. So with the type of knowledge that we are imparting them here, how to utilize it further is what is also important. And therefore, we always invite people. And once again, I thank you so much for accepting our invitation at short notice. And you have really taken the pains in making a very nice presentation. And in fact, it was also very informative.
Thank
So,
you.
yeah, that was about us. Now, are there further questions from
Yes, sir.
other
A couple of
participants?
questions are there, sir.
Yeah, please
Yes,
ask.
sir. A participant, Mr. Prithvesh, has a question. Prithvesh, if you could please unmute yourself and ask sir directly.
Yeah, please do.
Sir, in your starting slides, you asked about sesquiterpene, which is a precursor of artemisinin. So, what's the source of it?
No, sesquiterpene is an intermediate. So when you talk the biosynthesis of artemisinin, this sesquiterpene which I am talking about, is amorphadiene, is actually the intermediate, and that's what I explained to you, that how it is getting converted to artemisinin by further oxidations. So when,
And sir, this
if you really go back to the cholesterol biosynthesis, that possibly will clear you all the questions, because the mevalonate pathway is the part of cholesterol biosynthesis, and this one going in the next stage. And anyway, this is not done inside the human. I think this amorphodiene is not formed, but this amorphodiene is basically an intermediate for artemisinic acid and formed in the Artemisia annua, the plant which produces this product. Have I understood your question and answered correctly? Can you please tell me?
yes, sir.
Okay.
Yes, sir. We have another question on a different platform. That what are the different ways of using computational power, such as softwares and other type of such tools by which we do research in chemistry? So can you enlighten on this? That what are the industrial used such softwares, and what are their importance and current current system? Yes.
You see, computation and the computational science is, I think, a wonderful thing to know. I am really very poor in that. That's why I asked somebody to post the slides. I don't even know how to use computer. So I'm very bad, but I'm an old person. So for an old person, it's okay that I can bear with it. But for young people, I think they should learn all these techniques. And today, if we really keep our eyes open, then artificial intelligence and specifically the machine learning, which is the higher version of the artificial intelligence, it's really very important for people to understand that how it can be used. And I tell you that this is being used in drug discovery, in even designing synthesis and doing good chemistry. It is very predictive, and it gives a lot of good. Let me just tell you one thing. Whatever said and done, if you compare a human with your Google, do you have any idea about this Google Home or something which answers all your questions?
You're talking about the algorithm?
No, I am talking about the Hey Google. That if you, this is small, mic, and if you just ask question, it can answer questions without thinking of anything in the world. Means the questions are answered with such a quickness that even a human cannot even think in, maybe it goes to a literature search, it might take a month time. So the point is that whatever is provided by the technology, it should be taken as a useful, tool. And if you are interested in, and you can really click in Google, you will find out that these sort of techniques are very, very useful in chemistry, even designing synthesis, even designing drugs, even the discovery cases. And some of the systems are absolutely automatic, that you give them an idea, it automatically works, synthesizes compounds, tests the compounds, and also gives some results, and then again puts another hypothesis to do further sequencing on that. So I think this is very, very critical and very important, and particularly when you are in a technology area, it becomes even more important.
Also, sir, that was really a very the correct answer that we were trying to get. We have a couple of more questions in the comment section. Just a second, sir, will ask.
Yes.
So, yes. So I think there is another question which I'm asking from my side. Like
Please.
we have discussed about the Artemisinin.
Yeah.
There must be another compounds which Ipca might be working, or which your research group might be working, which might be derived from a green technology and green chemistry way. So can you please enlighten us regarding that?
No, you see, let me tell you that we are not per se working in green chemistry. As I told you that India is copying. So whatever is being done and whatever is required, and we are simply trying to make it better. So as I talk about my input, as I told you, that wherever we could make a difference, to make it simple, make it cheaper, make it better, make it more productive, more I think, lean and clean. These sort of things we have definitely done. But our objective is not green in true sense. So when we target as a green chemistry, no, we don't do, because I think it's not that easy all the time that everything can become green. So it is an important thing from the academic point of view that if you can work out, but luckily whatever work I presented to you, I think is almost 95 to 97% green. And even as Green Technology Award of the Europe, we actually reached almost at the end. I think we were only the last one or two that we could not get it. And this award which we got from the CPHI, this is a very, I think, valuable in true sense, because this is the conglomeration of all the industries, pharma industry in the world. They submit, all the industries are submitting their, whatever they have developed in a green aspect, and then it is scrutinized. So I think Ipca was the only company from India who actually won an award in the CPHI. So it was really a big achievement, and I think it was acknowledged that whatever work we have done, it makes some sense. So, but as far as the other products are concerned, I think there is not much time. But my main activity, I am not really doing now anything in the process research, which is a very, I think, normal routine. By you see the people, those they are now group leaders, they are doing much better job than me. I am more interested in thinking beyond that what we can do. So I am really more interested in the drug discovery. And we have done a lot of things. And just to give you a feel that one of the new molecule, we are now starting the phase 1 clinical trial. We are taking it to US FDA. And we are now going to approach again after doing some studies that can they even waiver in the phase 3 studies. One of the another molecule has been taken by MMV. You may not have heard the name. This is a malaria venture organization all over the world. This is a European organization. And this is the organization which takes care of any development taking place in the malaria segment. So development of new drugs in malaria or commercialization of that, redevelopment, whatever it is, everything I think is in their control. And one of the product is a new product again. We have a product patent on that. It was quite appealing to them. And now they have taken this. And this product is being developed by them. So they will be bearing all the expenses. I think it may cost around 40-50 crores at the minimum. And that is, I think, the clinical trial of that will be starting in May next year. We are at the development stage as far as the formulation and the API work is already done. Then there is another molecule which has shown very promising activity against the existing molecule on the pneumocystis pneumonia. That is again, I think that we have come up with the proof of the concept. And there are many more ideas, at least I have, which I am going to explore in various segments that includes anti-cancer activity and other things. But it's just not for the heck of it. It has a lot of substance in it, and the chances of success are very high.
Yes, sir. So there is one last question we would like to ask. There's a new biz, or I wouldn't say biz, there's a need of time, the circular economy, and how different industries tackle or say use this particular technological way to build their circular economy within their industry and with their customers.
Yes.
So how Ipca is trying to develop the circular economy and work towards climate emergency.
You see, if you talk about circular economy, it is covered in the green chemistry 12 principles actually. What it says that anything which you are generating as a waste, can you use it or reuse it? So that's actually the purpose of that anything which is coming out as a waste, can you reuse and make it a circular economy? Because what is happening? The consumer-driven developments, which we have till now. So you mass produce something, they do not work or they are no good for sustenance point of view after a year or so, then you throw them and it becomes a waste. And this waste is getting accumulated. And this is what we are talking about, that how the environment is getting affected. So circular economy is basically talking about that use green, this is circular economy, or make use of whatever is being generated as waste. So when I give an example, what I've just talked about, this is actually covering the whole aspect, that we are doing something where we are using the cheapest possible. You see why the raw material cost of this process is coming so low. It is using most of the green reagents, very cheap reagents, and the yields are very high. So when you talk about the first two principles of green chemistry, atom economy, this is that how much atom is really used and waste. Here what we have done, why I said that value from waste, this artemisinin which we are talking about today, based on Amorphadiene. When I was talking about to the CEO, when he told me that we are actually burning at this Amorphadiene. This Amorphadiene is being burned because we don't know what to do with it, so it's a waste for us. So when I heard this, I really thought that, okay, if this is a waste for you, can you supply some material to me and let me work on it? So you can consider it as an example of the circular economy or maybe the green technology. So it is actually the real use of the waste which they were burning, and they are still burning actually, to get it converted to product, which is the most valuable from any angle or any point of view.
Yes, sir, this makes totally sense. And I hope that in further lectures, maybe we are trying our best that as soon as our colleges will open and we get permissions, then we'll definitely, we would like to have you in our college, in our department, to have your live discussion on our platform in ICT.
Thank you very much. I am a regular visitor to ICT. I don't know how many talks I have given in ICT.
Yes.
Maybe I think dozens already I have given in chemistry department, because very regularly they keep on inviting me. So again, it's just a matter of chance that we have not met. But ICT is not a new place for me.
Yes, sir. Yeah, definitely, sir. Yes, definitely. We heard about that, what those visitors. So I think we are at the end of our session. Pritika, you may continue.
Yes. Thank you so much, sir, for patiently answering all our questions and for giving so much knowledge to us that we hope that we also endeavor to connect with you in the future. Thank you so much, sir. And with this, we conclude our session. Thank you, sir. With this, we conclude our session. I take immense pleasure in thanking our eminent speaker, Ashok Kumar, for such an informative talk and for contributing his valuable time. Thank you, sir. I would also like to thank Professor Vaidya, sir, for supporting us and for guiding us throughout all our events, and also for leading us in all our endeavors. Thank you, sir. Participants will be provided with a certificate of participation on successful submission of the feedback form, the link for which has been shared in the YouTube live chat box and also in the Google chat box, Google Meet chat box. E-certificates will be shared to the participants within 7 working days via email. I also thank all the participants who connected with us today and for being a very delightful audience. Lastly, I would like to thank the backend team of IPGT for managing the program. For any queries, participants can write us at the email shared on the screen. And thank you and have an amazing and productive day ahead. Thank you so much.
Okay, thank you. Thank you very much.
Thank you, sir.
Thank you, sir, for joining, and thank you to all the participants. We may now end session. You can leave on your on the time is right. Yes.